Kind of reproduce the chalk talk I did on osmosis and hypertonic, hypotonic, and isotonic solutions, just for anybody who happened to miss those days or who happens to need a refresher. So if we're talking osmosis, osmosis is the diffusion of water. And by the way, I am writing and speaking at the same time, so this video is going to be a little longer. Feel free to watch it at double speed if you need to. All right, so when we think of diffusion, we think of things moving from a higher concentration to a lower concentration. That's a little different with osmosis. Osmosis happens when the membrane that stuff is passing through is impermeable to your solute, but permeable to water. And I like to differentiate between solute and solvent. Remember, water is going to be your solvent. Water is the universal solvent. It dissolves a lot of things. And you have to differentiate that from what we're used to thinking of as concentration, which is our solute. Because remember, diffusion is where things move from where there's more of it to where there's less of it. So osmosis is basically moving from where there's more water to where there's less water. So let's look at a couple of solutions. So we have a couple of solutions here. We're going to assume there's equal amounts of water, etc. And let's say that I am putting in some solutes. I'm going to put different amounts of solutes in each of these solutions. And I'm trying to make it fairly obvious where the higher concentration of solutes is. Now, when I do this, I can't forget that there are solvent molecules in this as well. And I'm not going to draw the usual Mickey Mouse shape of water just for the simplicity of time, but we're going to assume, and we're just looking at proportions of these molecules, that these blue molecules are our solvent. And we're not counting absolute molecules, so not the absolute number of molecules. We're looking at proportions. So in the beaker on the left, we have a low solute concentration. And we have a relatively high solvent concentration. In the beaker on the right, we have a high solute concentration. And we have relatively fewer solvent molecules when compared. This is going to be kind of important to how I explain osmosis and those tonic solutions like hypotonic, hypertonic, and isotonic. So let's switch from thinking about beakers to thinking about a cell sitting in a solution. All right, so let's say that I have my solute molecules sitting in this solution. Some in the cell, some outside of the cell. So I am going to make a higher concentration of solute molecules inside my cell, and I'm going to put less solute outside my cell. Now I need to put in my relative concentrations of solvent. And again, this is not like I'm drawing every single molecule, especially for the interest of time. But these blue dots are supposed to be my water molecules. Now, if I look inside the cell, I have a higher solute concentration. I have a lower solvent concentration. Outside the cell, I have a lower solute concentration and a relatively higher solvent concentration. Now, if the solute was able to move across this membrane, because remember, this is a cell, Then it would move from where there's more solute to where there's less solute. It would move from the inside of the cell to the outside of the cell. And I'm going to represent that with, let's do a red highlighter. Okay? But it turns out that this membrane does not allow solute to cross. So let's get rid of that red highlighter. Instead, it only allows water to cross. So then we need to look at not what the solute concentrations are, but the solvent concentrations. Where is there more water? Outside the cell or inside the cell? Outside. Where is there less water? There's less water inside the cell. So we're going to have a relatively higher amount of water molecules moving to the inside of the cell. By the way, I want to go over some prefixes. These are prefixes for what I call the tonic solutions. There's hypotonic, isotonic, and hypertonic. And I always try to emphasize when I say hypo versus hyper. Hypo means less. If you are hypoglycemic, you have low blood sugar. If something is hyper, that means there's more. Think of hyperactive people. They're really active. So a hypotonic solution has less solute. Notice this is comparative. I'm talking less. Not that it has a lot of solute or an absolute concentration. I'm saying it has less solute. A hypertonic solution, on the other hand, has more solute. Again, note the comparative. I have more or I have less. You are always using these terms to compare two solutions. So while you might say, oh, that solution is hypertonic, it is understood that you're saying that solution is hypertonic compared to the solution inside the cell. And then isotonic, iso means equal. Think isosceles triangles, they have equal sides. So when you have an isotonic solution, there's going to be equal amounts of solute on both sides. All right, so let's look at this. Our solutions are, let's see, trying to do colors here. Okay, our solution inside the cell has more solute. It is a hypertonic solution. Our solution outside of the cell has less solute. It is a hypotonic solution. So notice water is moving from hypo to hyper. Let's see if that trend holds. So we're going to now have a different cell. Oh, before we move on, I want to point out, this is an animal cell. If a bunch of water is moving into it, what will happen to that animal cell? Well, the animal cell only has a cell membrane protecting it, and that cell membrane isn't super rigid, which means that it's got a little give to it. And if you start pouring more and more water into it, eventually it's going to pop. So what that means is that animal cells explode or lyse in hypotonic solutions. This is different if you have a plant cell. So let's say that I add a cell wall. That cell wall is relatively more rigid than the cell membrane. It is less flexible, has less give. And so while I can stick a bunch of water in it, that water is not going to cause that plant cell to burst. Instead, plant cells are turgid in hypotonic solutions. Turgid means they are completely stiff because they are completely full of water. More water can't enter once that cell wall has reached its limit for the amount of water it can hold. And so that cell is basically just kind of bursting at the seams, bulging with water. So let's say this was your plant cell originally. Yeah, hold on. Okay. Once you have a turgid cell, it kind of looks like it's bursting at the seams. So this is a turgid plant cell. This is the best condition for plant cells because remember, plants don't have bones. A lot of their structural support, not all of it, but a lot of it comes from the fact that the cell walls are helping to hold this plant up. And the cell walls can only do that if they are basically completely chock full of water. So turgid plant cells are best. Plant cells are going to be at their ideal condition when surrounded by a hypotonic solution. All right, let's look at what happens now when you have a cell in a different type of solution. So we're going to start with our animal cell, and our animal cell is going to be hypotonic to the other cell, to the outside solution. So let's start drawing this. And now got to add some waters, got a bunch inside, and relatively few outside. So let's talk solute concentrations and solvent concentrations. Inside this cell, I have a low solute concentration, but I have a relatively high solvent concentration. Outside the cell, I have a relatively high solute concentration and a relatively low solvent concentration. And remember, osmosis is the movement of that solvent. So it's going to move from where there's more solvent to where there's less solvent. So if we look at that, the water is going to move to the outside of the cell. So let's start talking in terms of hypotonic, hypertonic, etc. Inside the cell, there is less solute. Remember, less is the prefix hypo. Outside of the cell, there is more solute, so that is going to be hypertonic. Remember, hyper is more. So again, water moves from hypotonic to hypertonic. So if this was an animal cell, what would happen is animal cells, again, don't have a cell wall. They shrivel because all the water is moving out. And the fancy term for that, especially in red blood cells, is crenation. So they crenate in hypertonic solutions. Not good. So animal cells, they exploded in hypotonic solutions. They shrivel up in hypertonic solutions, neither of which is going to be good. Let's throw a cell wall around this and make this a plant cell. Plant cells still going to lose water in a hypertonic solution, but we have a fancy word for that. It's called plasmolysis. And plant cells, we can say they plasmolyze in hypertonic solutions. And this, when you look at the actual ultimate cell, not only does it lose a lot of its water, but it loses so much that the cell membrane starts to pull away from the cell wall. And so you end up with what kind of, according to y'all in class, looks like a starfish inside of a box. I know I drew the wrong number of, quote, arms for a starfish, but this isn't actually a starfish. So the plant cells that are in a hypertonic solution are basically dead. There's no way they can come back from that. So don't be like Miss Mystery and don't let your plants get to the point where they have hypertonic solutions around their cells. So this is bad for plants. This is bad for animals. We still haven't found a condition that's good for animal cells. So let's go back to the drawing board again. Now, oops, wrong thing. I'm drawing relative concentrations. So remember, this isn't like 100% accurate. But what I'm trying to represent is the fact that there are equal concentrations of solute inside this cell as outside this cell. That means the relative concentrations of solvent are also equal inside and outside the cell. So that means that you're not going to have water entering the cell or leaving the cell. Now, does this mean that the water's not moving at all? No. What this means is that for every water molecule that moves inside the cell, another one is going to move outside the cell. This means that there is no net movement of water. That net means there's no total movement. There's no change in concentrations. For every bit of water that enters the cell, the equal amount exits. This is a system at equilibrium, meaning it's not going to change. Now let's talk tonics. Remember, iso means equal. So we have equal concentrations of everything inside and outside the cell. That means the solutions are isotonic to each other. So animal cells, they don't have to lose water. They don't have to gain water. They are normal in isotonic solutions. So for the vast majority of your cells, the solution concentration inside the cell is equal to the concentration outside the cell. You don't want the cells to lose water. You don't want the cells to gain water. This is the ideal or best condition for animal cells. All right, now we throw a cell wall around there, turn this into a plant cell. Plant cells are not going to be turgid. They're not going to be completely chock full like they would be in a hypotonic solution. So we say they are flaccid. Flaccid means kind of limp, means that it's not completely, totally full. And that is not healthy for a plant cell. So this would be if your plants have an isotonic solution around them, then they are going to be wilted, meaning they're not going to be like fully healthy. All right. And then the ultimate thing to remember, water always moves from hypo to hyper. Always. Memorize that. Water always moves from a hypotonic solution to a hypertonic solution. I also think of things as water moves. Let me write that with a little thinner pen. From the less concentrated solution to more concentrated solution until the concentrations balance out. Basically, until the system reaches equilibrium. All right. Hopefully that helped you with osmosis. I know this was a difficult topic when I myself was learning this in high school. So hopefully this works for you.